And I think next is the 39-fold symmetry of the Wald protein. So one subunit is seen as a red thread here. And here, seen from top, you see one subunit here. And the single subunit is a very funny one, starting with amino acid 1, and there's 1, 2, 3, 4, 5, 6, 7, 8, 9, essentially identical domains, and then comes a green domain, and then a long helix, and then something at the end. So proteins can have an enormous variety of shapes and symmetries. And there are also proteins with multiple symmetry axes. So, for instance, there's a 2-2-2 symmetry. So hemoglobin, for instance, is a tetramer with the 2-2-2 symmetry. There are 2 times 2 different subunits here. And there's 3-2 symmetry of aspartic carbonyl transferase. So there are 3 times 2 times 2 subunits. So there's, say, hexamer arrangement in 2 layers. The Meridian or glycolate oxidase has 4 subunits in 1 ring, and there are 2 of those rings, so it has 4-2-2 symmetry. And glutamine synthase has 6-2-2 symmetry, and ROE-EL that I talked about before has actually 7-2 symmetry. So there are 2 layers of 7-fold symmetry. And then whole range of different symmetries, 4-3-2 octahedral ferritin, 24 subunits, and the icosahedral viruses, which have 5-3-2 symmetry. And I think I'll show you a picture of how they look. So they have 60 times 10 different subunits. So here's transliterating, and here's a virus structure, the coat of a virus. So this is flavivirus, which has the icosahedral 5-3-2 symmetry. And let's see if we can identify these. The subunits, the green, the red, and the blue subunits, they are chemically identical. They have exactly the same amino acid sequence, but they have slightly different shapes. So the blue ones are sitting around a 5-fold symmetry axis. The red ones are sitting around the 3-fold symmetry axis, and the green ones are sitting around the 2-fold symmetry axis. And they all make an icosahedral arrangement in this beautiful way. And as you know, the virus nucleic acid is kept inside here, protected by the protein shell. Then we have the helical symmetry. So actin, for instance, the muscle protein actin has helical symmetry. And the helical rise is 27 angstrom per monomer and 167 degrees. So not a half turn, but almost. And the pilus structure, type 4, has different arrangement. The filamentous phage has different arrangement. The Tobacco mosaic virus has a different arrangement, but they all have helical symmetries. So here's, for instance, actin. We see blue subunit, red subunit, yellow subunit, and green subunit, and they all go in a helical way. You can add further actin molecules in this direction by the same arrangement. Here is tubulin, and here the 2 different subunits, the alpha tubulin and the beta tubulin, and they also make a nice helical arrangement in this way. So aggregation is sometimes a natural thing in proteins, but sometimes it's also not natural. And many proteins can aggregate upon denaturation. But certain proteins are aggregated in vivo and cause diseases. And I'll show you a number of those. But first, an example from simpler, like silk. this aggregated protein. And I think Indonesia is a silk producing country, so silk must be important for you. And this is a diffraction pattern of silk. So you can see that it doesn't form really nice spots, but certainly a number of interesting features here. And silk has repeat sequences of glycine, alanine, glycine, alanine, glycine, and serine, or maybe a tyrosine at the end here. And the arrangement in silk is something like this. So this is a beta sheet. Each of those is a beta sheet that goes down through the board. And the beta sheets are packing very beautifully on each other. So the method group here of the alanines is pointing into the slight cavity here, and this method group is pointing into this cavity. So when there's no method groups in between, they come very much closer, and when there are method groups between, they are a little bit more further apart. So this is the simple structure of poly-glyala. So every second residue is glycine, and every second is alanine, which is the imitation of the silk. But the protein misfolding disorders are numerous, and you know them from the medical field. Alzheimer's disease, there's a protein or a peptide called the A-beta peptide. Creutzfeldt-Jakob's disease, there's a prion protein which is aggregating. Parkinson, alpha-synuclein, Huntington's disease, Huntington, type 2 diabetes, IAPP, familial amyloidotic polyneuropathy, triglyceride, and the same as senile systemic amyloidosis, and so on. There's a whole range of this type of misfolding disorders, which are terrible when they happen to you. And say, in some cases, it's a very classical protein called, in this case, lysozyme. But why and how do they aggregate? So this is the diabetes type protein, IAPP. Here you see the fibers formed by the protein. And this shows a very classical pattern. If you take the fibers and take an X-ray photographs or photograph of them, you see certain very elevated features. You see a ring out here which is corresponding to repeated distance of 4.7 angstrom, and you see then in a particular direction a distance which corresponds to 8.3 angstrom. So this is the classical cross beta diffraction pattern, and this cross beta is say primarily observed by diffraction experiments. So what happens here is a certain peptide of the protein is bound to form a beta strand, and these beta strands have a tendency to hydrogen bond to each other. So strand after strand, identical strands here are lined up with each other, forming an aggregate. And this is part of the problem. And then another side of the whole thing can be this domain swap, such that the peptide here can be the connecting part between domains, and then the domains can form this domain swap arrangements, such that you make a very large aggregated structure, which is not what the protein was meant to do. So, for instance, one structure which has been investigated is SUP35 from yeast, and this is a prion protein, and the sequence glycine, asparagine, glutamic, asparagine, tyrosine causes the aggregation, and it forms the very tightly packed aggregates. So here you can see the arrangement between the beta strands, And you see the hydrogen bonds between the peptides, the peptide planes, and also between the side chains. So they seem to be beautifully designed to be organized in this way. And if you look at, so here's then a whole set of sheets which go in this way, and then they have a neighboring set of sheets below, and they make again a very beautiful package. So the side chains here of the different layers pack beautifully into each other and make the hydrogen bonds needed. And then on the next step down here, there's not equally perfect arrangement, but here water molecules fill up the empty spaces, and again below here you have the same arrangement as there. So the whole thing becomes a very, very well designed and packed structure. So let's leave that and now come to some other aspects. Proteins also have prosthetic groups, coenzymes. They need these groups to be able to function properly. They can be heme, they can be chlorophyll, NAD, NADP, S-methionine, they can be metals. So the classical heme group is looking this way, and you know, chlorophyll is very close relative of heme, but it has some magnesium sitting in the place of the iron. So many proteins bind metals. Some of them bind them very strongly, some bind them very lightly. And sometimes these metals stabilize the structure of the protein, and often they're also part of the function of the protein. So there are numerous types of metals which have been found in proteins. So there are certain metals which prefer oxygen. Sodium, potassium, magnesium, and calcium, they prefer definitely oxygen ligands. And here you see the number of ligands they usually have. Sodium 6, magnesium has 6. And what type of residues are binding here, and the distances which you see. So for instance, the potassium channel that I showed you before lunch had this 2.8 angstrom distance between the metal position and the carbon and oxygens. Sodium would like to have 2.4, but sodium will not then fit into this potassium channel, because it would like to have the ligands in a different way. But then there are also metals with variable ligand preferences, like manganese, iron, nickel, copper, zinc, and they have different oxidation states, they have different number of ligands, and these are the ligands they usually are found with, and these are the distances between the metal and the ligands. And they have different roles. As you know, zinc, copper, nickel, iron, and manganese, they all also have very specific functions in various proteins. So in many cases, the positive charges of the metals are neutralized by the ligands in different ways. So I'll show you some examples, and this is magnesium ion bound to ATP molecule. So here's ATP, and this magnesium is bound between the beta and gamma phosphates. And you can see here that the magnesium is very particular with regard to the orientations of the ligands. So these 4 ligands here, they form a distinct quadratic plane, and then there's one ligand from above and below, and it would like to have as perfect as possible octameric arrangement, otherwise forget it. Magnesium is very selective for octahedral environments. here's calcium, and calcium is much more flexible. It doesn't really care. So the number of ligands doesn't matter so much, and which way they come is also. So here you see 1, 2, 3 carboxyl groups. You see ligand from the peptide chain, and here's a water, and here's a side chain. So this ends up being 7 ligands, I think. Here is cobalt bound to the choline ring and additionally histidine and 1 methyl group here. So this is methionine synthase. And irons can be of different kinds. Here you see an iron atom bound to 4 cysteines in a protein. But iron can be also more complicated. Like here, we have a so-called iron 4 sulfur 4 cluster. So this is a cluster of irons which are forming a cubic system, but every second position is filled by a sulfur atom, inorganic sulfur. And here you see this cluster sitting in a protein. So these type of inorganic clusters are also found in quite a number of proteins, sometimes very complicated structures. Here's nickel in the enzyme urease, and these are 2 nickel atoms and bound to 3 histidines, aspartic acid, and I don't know what this is. This is probably a ligand here, and 3 water molecules. Now, this urease I'll talk about, I think tomorrow, or no, on Wednesday, because urease was actually a very critical protein in making it clear that enzymes are proteins. And there was a person called Sumner who made it very clear. He purified urease activity. At that time it was not